James Biology. Previously we have discussed about structure of polynucleotide chain and Chargaff's rule and other basic things about molecular basis of inheritance. Today we are going to discuss about Watson and Crick model of DNA. Okay, Watson and Crick proposed the double helical structure of DNA on the basis of X-ray diffraction produced by Maurice Wilkin and Rosalind Franklin in the same year, that is in 1953. On the basis of that X-ray diffraction, Watson and Crick proposed the double helical structure of DNA. Okay, let's see what Watson and Crick proposed about DNA. So, James Watson and Francis Crick in 1953 proposed the double helix model of DNA. Double helix. Double means two. Helix. Helix means what? Helix means spiral. Helix is a Greek word which means spiral. Spiral or twisting. Okay, helix means spiral or twisting. So, when any surface wrapped around a cylindrical surface, that is called spiral or helix. Okay, so helix means spiral. James Watson and Francis Crick in 1953 proposed the double helix model of DNA. Let's see the features what Watson and Crick proposed. DNA is made up of two polynucleotide chains. We have already discussed it that DNA is made up of two polynucleotide chains. You can watch the previous video for better understanding about the polynucleotide chain. The backbone is consisted by sugar phosphate. The backbone we have already discussed in polynucleotide chain. This is the backbone. This is one strand. I have drawn here red color in one strand and green color another strand. The backbone is consisted by sugar phosphate while nitrogen base project inwards. These are the nitrogen bases. Adenine, guanine, thymine, cytosines all are nitrogen bases. The nitrogen bases are project inwards. Suppose this is sugar. Okay. And here the 5' carbon, here one phosphate group is added. And again this is 3' or 3' end of one sugar. This is sugar. Here OH group is present. And here another phosphate is added to the 5 carbon compound of another sugar. Okay. Again here 3' and a phosphate group. It is added to another 5 carbon of another sugar. So backbone is formed by sugar and phosphate like this. Okay. And the nitrogen bases are added to the first carbon atom here, nitrogen base. Suppose thymine. Okay. And it is added to the another sugar. Here 5, here also phosphate, and again like this phosphate empty, add he. Okay, the nitrogen bases are project inward towards inward side. If thymine is present here, it will added to the adenine of another sugar of another strand. Okay, We have already discussed it. You can watch the video for better understanding. Okay. Let's come to the next point. Two chains run in anti-parallel fashion with 5' to 3' polarity or 5' to 3' polarity in one and 3' to 5' polarity in another chain. Two chains are run anti-parallelly. Okay. If one strand starts from 5' to 3', another strand will start from 3' to 5'. Okay. Polarity. Polar means direction. Okay. The direction of two strands are opposite. Okay. If one strand starts with 5', another will start with 3'. Okay. The bases in two strands paired through hydrogen bond. Bases. Bases means nitrogen bases. We have studied here that the nitrogen base project inwards. Here the nitrogen bases here cytosine, adenine, thymine, guanine. Okay, in one strand and other strand guanine, thymine, adenine, etc. And these two strands are attached together through hydrogen bond. Okay, the bases in two strands pair through hydrogen bond, and hydrogen bonds are formed between the nitrogen bases. Okay, adenine bonded with thymine with the help of two hydrogen bond. Adenine always added to thymine by double bond, double hydrogen bond, and guanine bonded with cytosine by 3 hydrogen bond, and guanine always bonded to cytosine with 3 hydrogen bond, and vice versa. Okay, adenine can attach to thymine through 2 hydrogen bond, and vice versa. Thymine can bind with adenine with 2 hydrogen bond. Like this, guanine bind with cytosine with the help of 3 hydrogen bond, and vice versa. Cytosine can bind with guanine with the help of 3 hydrogen bond. Okay. Next point. Two chains are called right-handed fashion. Two chains are called right-handed fashion. Right-handed fashion called ହେଇକି ରହିଛନ୍ତି ଗୋଟେ cylindrical plane ରେ. Helix means ଗୋଟେ cylindrical plane ରେ ଏମାନେ କଣ ହେଇଛନ୍ତି ଏମିତି right handed manner ରେ spiral ବା coil ହେଇକି ରହିଛନ୍ତି କିଏ two strand ବା double stranded DNA. Okay, the pitch of helix is 3.4 nm. Here the pitch of a helix, double helix structure of DNA is 3.4 nm. What is pitch? Pitch means 1 helical turn. The distance between 1 helical turn. Okay, गोटिए हेलिकल टर्न नेला बेलकू जतिकी डिस्टेंस कवर करूची ताकू कह जाउची पिच, एंड द पिच ऑफ डीएनए डबल हेलिक्स इज 3.4 nm. Okay, and one helical turn contains 10 bases. Here the number of base pairs in each turn is equal to 10. Okay, one helical turns contains 10 base pairs. Okay, the double helix of DNA turns after every 10 base pair. Okay, if one helical turn can complete after pairing 10 bases. Okay, 10 nitrogen bases. Let's count here between the pitch. Pitch means one helical turn. Here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For example, okay, so one pitch or one helical turn contains 10 base pairs. 10 base pairs. Okay, a double helix turn after pairing 10 bases. Okay, clear? Next point. The distance between two adjacent base pair. Distance between two adjacent base pair is equal to 0.34 nm. Okay. One base pair stuck over the other in addition to hydrogen bond, which provides the stability to the double helix. One base pair stuck over the other. Here, GC stuck over TA stuck over CG. Okay, one base pair stuck over other in addition to hydrogen bond, which provides the stability to the double helix. Okay, the stocking of nitrogen bases in addition to hydrogen bond provide the stability to the double helix of DNA. Okay, we have studied that the backbone consists of sugar and phosphate. It is very important. The backbone of a double helix structure of DNA consists of sugar and phosphate, and the stability provided by the nitrogen base in addition to hydrogen bond. Okay, जदि मझिरे हाइड्रोजन बॉन्ड न थांता तो एठी सेमति रहिला तार नाइट्रोजन बेस गोटे स्ट्रैंड रे एपटे गोटे स्ट्रैंड रे नाइट्रोजन बेस रहिला तो दीटा जाको कोन ऐड हे परले सेपरेट रहिले तो दीटा को स्टेबिलिटी किए दोची हाइड्रोजन बॉन्ड दीटा नाइट्रोजन बेस को दीटा स्ट्रैंड रो दीटा नाइट्रोजन बेस को ऐड करिकरी स्टेबल दोची बा स्टेबिलिटी दोची के हाइड्रोजन बॉन्ड ओके सो दिस इज Watson and Crick model of DNA. And very important point or object of Watson and Crick model of DNA is the complementary base pairing. Complementary base pairing. What is the meaning of a complementary? Complementary base pairing. Complementary. What is the meaning of a complementary? Complementary means related with each other. परस्पर सहित संबद्ध बा परस्पर सहित परिपूरक को माने the nitrogen bases. Okay, very important objective of Watson and Crick model of DNA is complementary base pairing. It means if we know about the bases of one strand, we can easily predict the bases of another strand. Because guanine always can add to cytosine, thymine can add to adenine and vice versa. That's why if we know about the base of one strand, we can predict easily the bases of another strand. Okay, so this is Watson and Crick model of DNA. James Watson and Francis Crick in 1953 proposed the double helix model of DNA on the basis of the X-ray crystallography or X-ray diffraction produced by Wilkins and Rosalind in the same year in 1953. जेमती आमे डॉक्टर को पाखु जेतोले जाउछु जदी हम हाथ गोड़ कोन जगह पेन होची आमे प्रथमे जाई कि एक्सरे करूची तो से एक्सरे को बेस करी कि डॉक्टर हमको प्रेस्क्रिप्शन दउचंदी बा किछ स्टेटमेंट दउचंदी ठीक सेही भली विल्किन और रोसलिन को एक्सरे डिफ्रैक्शन को आधार करिकी वाटसन एंड क्रिक प्रपोज करिथिले डबल हेलिक्स स्ट्रक्चर ऑफ डीएनए ओके सो से कौन कहिथिले ना डीएनए इज मेड अप ऑफ टू पॉलीन्यूक्लियोटाइड चेन डीएनए दुईटा पॉलीन्यूक्लियोटाइड चेन को लेके गठित द बैकबोन इज कॉन्स्टिट्यूटेड बाय सुगर फॉस्फेट जो बैकबोन अछि मेरुदंड कोथरे तयारी होची ना सुगर और फॉस्फेट sugar phosphate एमती एडिशन हेकी तर बैकबोन क्रिएट होची व्हाइल नाइट्रोजन बेस प्रोजेक्ट इनवर्ड्स जो कि कोन होची नाइट्रोजन बेस गुडा को भीतर आड़ को प्रोजेक्ट हेकी रहउचंती टू चेन्स रन इन एंटी पैरेलल फैशन गोटे डायरेक्शन में पड़ुथला आ गोटे डायरेक्शन में पड़ु होची सेति पई कि कोन होचंती दे आर एंटी पैरेलल ओके जदि गोटे स्टार्ट होची 5 डैश टू एंड होची 3 डैश रे आ गोटे स्टार्ट होची 3 डैश रे एंड होची 5 डैश रे ओके with 5 dash to 3 dash polarity in one and 3 dash to 5 dash polarity in the other chain okay and another thing is we can calculate the percentage of nitrogen bases by this formula A plus T is equal to 100 minus G plus C. It means A plus T is 50%, G plus C is 50%. Okay. A plus T is equal to 100 minus G plus C. We can calculate the percentage of nitrogen bases through this formula. Okay, which is proposed by Watson and Crick. Okay, next point. The bases in two strands paired through hydrogen bond. Bases nitrogen bases are paired through hydrogen bond. Dita nitrogen base होची pair होची hydrogen bond हेगी ना नहीं दीटा स्ट्रैंड रे दीटा नाइट्रोजन बेस प्रेजेंट होउची ते हाइड्रोजन बॉन्ड क्रिएट हेकी दीटा जाको स्ट्रैंड अटैच होउचंती adenine bonded with thymine with the help of two hydrogen bond adenine bonded with thymine with the help of two hydrogen bond and guanine bonded with cytosine by 3 hydrogen bond next two chains are coiled in right-handed manner or right-handed fashion. Okay. Pitch of the helix, one helical turn is equal to 3.4 nm or 3.4 into 10 to the power minus 9. Okay. And each helical turn contains 10 base pair. Okay. Number of base pairs in each turn is equal to 10. And the distance between two adjacent base pair is equal to 0.34 nm. Okay, one base pair stuck over the other in addition to hydrogen bond which provides the stability to the double helix. Okay, and the diameter of a double helix DNA is 2 nm. Okay. So, this is all about Watson and Crick model of DNA which is very easy to understand and very easy to write also. So, in the next class, in the next video, we will discuss about the packaging of DNA and the central dogma. After that, replication and transcription translation. Till then, keep watching and keep learning. Don't forget to subscribe my channel, that is AN's Biology, and follow me on Facebook for more videos. Thank you.